Hello, my dear children. Namaste to all of you, and very warm welcome to yet another super amazing session of the much loved, very popular Sprintex series for ICSE. This is Ambika, your biology master teacher, right here on this super duper amazing platform of Vedantu, which is Vedantu 9th and 10th English. Alright, guys, so as you know, Sprintex is a series where we intend to give you that extra winning edge. Basically, we're going to be discussing a lot of questions, either based on the pattern or some of them are past year's questions themselves. Alright, but basically, the idea is to help you face questions which are with a lot more confidence than ever before. All right, now let us move ahead. We've already done a couple of chapters. Today we are going to discuss questions from the chapter Absorption by Roots. All right, let us move ahead. So in this chapter, these are the major topics, okay? Imbibition, diffusion, and osmosis, plasmolysis and deplasmolysis, absorption of water and minerals, active transport, passive transport, and ascent of sap. So children, now those of you who are new here to our channel, let me remind you something. First of all, this Sprintex series is not intended to give you a basic understanding of all the concepts in the chapter. As I told you, as I've made it very clear, it's going to be question answer discussion that will be happening in this series. All right, for those of you who want to learn every single concept from all of these chapters well in detail, don't worry at all. In case you are new to our channel, please check out all the playlists out there on the channel's main homepage, and you would find all our different playlists: the Umang series, the one shot series, mind map series, VIP series, now Sprintex series, so many things. There's also Bio Bites in 15-minute series. So many useful and super interesting sessions that you can have a look at. Some of them, many of them were live. In fact, some of them were premiered also, but that doesn't matter. Now, when you want to learn, it matters that it's still available to you and it's there. So please have a look at it. Make the best use of it. All right. And to make sure you don't miss out any of our sessions in future, I would say the best way is to stay subscribed to our channel. So click on the subscribe button right now, which is right below this video, and click on the bell icon right next to it so that you get all the notifications. Because on this channel, children, we put up everything which will help you, which is enough to complete the preparation of CBSE 9th, ICSE 10th, and CBSE 10th students. And I would say even students of other grades, lower or higher grades, can find a lot of useful material here, because we do a lot of general motivation and tips and tricks and study strategies and so many other things also that will benefit everyone. So please stay subscribed and also share word about our channel to all your friends, because we completely are dedicated to this. So what do you think we would want in return? All that we want is that maximum number of children in India and across the world should benefit from this channel. All right, guys. So let us move ahead. Remember, I had asked you a question as homework. Those of you who were with me in the previous session, that is, give reason. Children of the same parents, howsoever similar, are different from each other in certain aspects. Why do you think so? Well, I've put answers of Pranjal Yadav and Bhavya Priti here. So Pranjal says the children of same parents are different from each other in certain aspects due to the mixing up of genes from mother and father in different ways. Very well said. And Bhavya has phrased it this way: children of same parents are different from each other in certain aspects. I guess it's aspects due to differences in gene patterns during crossing over, which results in genetic variation. So I think both of them, Pranjal and Bhavya, have mentioned two crucial points here: crossing over resulting in genetic variations. And Bhavya has also added, it was a very amazing session. Thank you so much, Bhavya, and all the other children who've also given me the answer for this particular question. Thank you very much, children, because it means a lot to me, especially in premiered sessions when you take off a few seconds or a few minutes of your time to give me the answer. It shows how dedicated you are and how you want to be involved in interacting with us. And that feels amazing. Thank you, children. Thank you so much. So, yes, let us have a look at your biology schedule. I've been sharing this with you, and your other teachers have also been sharing this with you. ICSE 10 students, please make a note of this. You may take a screenshot or you may download the PDF, which is available in this description box below, whatever works for you. But please make sure this is the timetable that you know about. Okay, note it down somewhere so that you don't miss it out. So as I told you, best way, easiest way is staying subscribed and clicking on the bell icon. Okay, so as you can see, the biology sprint text schedule is right here. We started off on the 8th of Feb. We are done with the first 2 chapters, and we are today in this. Okay, absorption by roots. So these chapters are remaining. On some days we have sessions at 5 p.m. On some days it's going to be at 6 p.m. And we will be ending the sprint text for ICSE on the 27th of Feb. Note down the date, which means by 27th we are going to be done with all your chapters. Right. Chemistry, Anubha ma'am also has an amazing schedule for you, and ma'am has also already started off. Some of your topics are already done, and ma'am will continue to be doing sessions with you at 6 p.m. on these dates. So ma'am is also going to be finishing on the 27th of Feb. Then this is your physics schedule by Anup sir, and Anup sir has also already started off, and sir will be ending your syllabus on the 26th of Feb, as you can see, 5 p.m. I think every day it's at 5 p.m. And math schedule is right here for you. Gopal sir has also already started off, and sir has a lot of sessions to do, and this will be ending on the 26th of Feb. Gopal sir sessions, all right. And then comes Shweta ma'am's sessions. So here is Shweta ma'am's English schedule for you, right here, children. And this will also go on till the 27th of Feb. And comes Ankana ma'am's social studies schedule. So I think Gopal sir, Shweta ma'am and Ankana ma'am probably have to do many more sessions as compared to us science teachers, especially this year. So you can see that the social studies topics are all right here. Oh my goodness. Yes, so many sessions that Ankana ma'am has scheduled for you till the 27th of Feb again. All right. So note down the time also, because Ankana ma'am has got different timings, 9 p.m., 6 p.m., I think 7 p.m. also on one of these dates. So make sure you note down this timetable somewhere for all your subjects. We are going to be finishing your sprint text series by the 27th of Feb. So children, let us move ahead, and remember to stay subscribed if you haven't done it yet. Click on the like button right now to show me your love for sprint text series, because all of you kept asking me for it, right? So that's exactly why we started off with sprint text for ICSE right here. Right, children, the happier you are, the more beautiful you become. Always remember this. Yes, now getting started. Question number 1. Name the instrument used to measure root pressure. So it can come as a 1-mark question. Sprint time, well, I would say not even 30 seconds. Ideally, you should be able to answer this within 15 seconds. The answer is manometer. The instrument used to measure root pressure is called manometer. 1 full mark if you just know this simple concept. Righto, moving on to question number 2. Name the solution, or rather the type of solution in which the cell swells up. So basically the question is, you are placing a cell in a solution and the cell is swelling up. What kind of solution was that? Yes. Think about all that we've learned related to this, and your time ends in 3, 2, 1, go. The answer is, of course, hypotonic. You place a cell in a hypotonic solution. Hypotonic solution would mean it's an entirely dilute solution with very, very little solute concentration in it and a lot of solvent molecules in it. So solute concentration is going to be greater inside the cell as compared to the outside, and as a result, water is going to be entering inside the cell. Water is going to be entering inside, resulting in swelling up of the cell. So hypotonic. Remember, hypertonic would have a high solute concentration, isotonic would have equal concentration as that of the cell. Hypotonic would have lower solute concentration as compared to the cell. Okay, so again, simple question to fetch you 1 full mark. Let's move ahead. Question number 3. Give the scientific term for sticking of water molecules to the surface of a cell. Sticking of water molecules to the surface of a cell. Yes, how is this relevant? Where have we learnt this? Remember? Okay, your time ends in 3, 2, 1 and go. The answer is adhesive force. Remember this, the sticking of water molecules to the surface of a cell, and between adjacent water molecules also there is a force of attraction. Do you remember what you call that? Okay, think about the answer. Put the term, if you remember it, in the comment section below or in the chat box. I would love to have a look at it while the session is getting premiered. All right. Yes, children. So this is something that we learned in the concept of ascent of sap and maintaining the capillary action and all of those. All right. So again, 1 full mark is completely yours. Question 4. Active transport needs energy, osmotic pressure, root pressure, or all of them. Active transport, active transport. Your time ends in 3, 2, 1 and go. The answer is energy. Always remember, active transport would need energy. Passive transport wouldn't require energy. No energy requirement. Okay? Right. So remember this. Active. Very, very active because energy is involved over here. All right. All good? Okay. Moving ahead. Question 5. Most plants have a bunch of root hairs. Give reason. What do you think could be the reason for this? Bunch of root hairs. Well, absorption by roots is the chapter's name. Why do you think roots? So I'll use a different color to just mark off something. Okay. Root hairs. What do? Children, remember the plant body and these are the root hairs. Right? So there would be very, 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 very tiny, not even as big as this. They would be really tiny root hairs which are over there on the surface of the roots. And root hairs increase the surface area for water absorption, just like there are villi in our small intestine, right? Villi, finger-like projections in our small intestine to increase surface area for absorption. Root hairs increase surface area for water absorption in roots. And also remember, rate of water absorption is proportional to the rate of transpiration. Keep that in mind. Okay? Right. But anyway, since the question carries 1 mark, I think even just the first point would do. The second one is there as an additional supporting statement. All right. So we are done with 5 questions carrying 1 mark each. Children, what do you think about it until now? Are you improving? Are you learning something? Yes, any questions, any doubts that you have, please, please, please feel free to post it in the comment section below. All right. Yes. Now, what is transpirational pull? Again, something that could either come as a standalone 1 mark question or something that could come for more marks as a part of a 5 marks question. Okay. Now remember, in transpirational pull, what happens is, if this is the plant body, okay, these are its roots, this is the soil, okay. What happens is, during transpiration, water gets lost in the vapor form from the leaves, right, from the aerial parts of the plant body. And this decreases the water potential. So this creates a gap basically because water molecules are getting lost. So because of cohesion of water molecules, attractive force between adjacent water molecules from below the water column get pulled up. This pulling force is called transpirational pull. You could add, in case the function is asked, you could add that it adds to or it aids in transport. It aids in water and mineral transport in a plant body. Okay? Right. Okay, so that was question number 6 for you. Now time to take a very quick break because I need to tell you something that's going to help you gain 100% knowledge and as a result score 100% marks. What is it? Many of you may aware of it. Unlimited live classes with fun and high level quizzes. Yes, children, I am talking about the courses we have at Vedantu through which you can compete with students across the world. And interactive replays with live quizzes and leaderboards, because some sessions, when you miss them happening live, you can always catch up through the replay. But the interesting thing is, quizzes and leaderboards are still going to be live over there, right? Okay, so on top of this, you can also download premium handwritten notes of your master teachers after every single session, and doubt solving, session assignments, tests, chapter assignments, everything which is designed exclusively to help you feel better, feel much better, confident before you appear for your board exam, before you appear for your final exam. All of this comes as part of this, children. And also on top of this, icing on cake, as I would love to call it, 5,000 plus topic wise or chapter wise micro courses and free crash courses for competitive exams. When I say free, it's for those of you who are registered for Vedantu Pro subscription, and that's not a big deal because it's going to be available to you at a very, very reasonable cost. And I will tell you about it, because more classes would mean lesser price. How exactly? Well, children, to find out more about this, just visit the link given in the description box below, okay, or the pinned comment, and just apply the coupon code AMBPRO, okay. Now, for a 1 month subscription, which means in 30 days, you get unlimited access to your classes of English, social studies, mathematics, science, and that's about it. So all your 4 main subjects. Well, I haven't broken down these into biophysics, chemistry, history, civics, geography, or anything like that, but everything comes under this same umbrella. All right, but 4 major subjects for you, which are going to be priced at 2,699, everything together. Okay. But even on top of this, just apply a coupon code AMBPRO. Note this, AMBPRO. All right. So this will be available to you at just 2,159. Can you imagine? The entire course for you, which means approximately 200 live sessions, if not more. For these many sessions, you pay just 2,159. And that means your per session cost, do the basic math, comes down to rupees 11 per session. Isn't that amazing? Because this is almost equal to a regular lace packet. Can you imagine, children? Well, not just a lace packet, I would say. It's even more reasonably priced than, because I'm a chocoholic, I like to think about it in terms of chocolate, even more reasonably priced than a decent chocolate bar, right? A chocolate bar that would give you satisfaction of having eaten it, right? So this is even more reasonably priced than that. And imagine knowledge being available at such an amazing price. That's only because Vedantu is dedicated to helping you come out with flying colors. So join us. Don't hesitate to check out more details. All right, guys. So let us continue. Question number 7. How are root hairs adapted for absorption of water from the soil? So about the root hairs we've already discussed, but more about it. How exactly are they adapted? They are thin-walled. Okay. Then concentration of cell sap of root hair is more than soil water. How do you think that is being helpful? Remember, children, inside a root hair cell, inside the cell sap, okay, the sap of the root hair cell, concentration of the solutes is going to be really, really high as compared to outside. As a result, what do you think? As a result, the outside environment is always maintained as a hypotonic environment. And as a result, water would be drawn in from outside to inside naturally by osmosis. Water naturally enters by osmosis. So in order to maintain this within the root hair cell sap, there is always the maintenance of a high solute concentration. This is the main point for this. And of course, they are thin-walled as well. Okay? Right. So children, these are the points which can easily fetch you 1 mark, if not 2, Easily it can fetch you 1 mark. Okay, so children, you know what it's proving? Like what I'm able to see is that uh, these questions, as a student, I'm not thinking about it as a teacher. If I think about it as a student, these questions are actually going to be helping me identify my strengths and my weaknesses in this particular chapter, absorption by roots. Isn't that amazing? So children, do this simple exercise. Wherever you think you are lagging behind by understanding of these questions in this series, make sure you go back to our Umang series or any of our other series. I would suggest Umang because that's where we discuss everything in detail, right? Check it out. Go to the particular session video and please brush up your concepts. Maybe you might have to watch it several times. Maybe you would have to read through your textbook several times. That doesn't matter. Now is the time to improve upon your weak areas. All right, not on the eve of your exam. Okay, now question number 8. Explain the term plasmolysis. Give one application of this phenomenon in our everyday life. Children, now that reminds me, when I look at this question, another way of asking this question exists. You know how? It could come as a part of a 5 marks question in section B. You could be given a diagram of plasmolysis. You might be asked to identify the phenomenon represented in the diagram. The second part of the question could be, give one application of this phenomenon in our everyday life. And there might be a third, fourth, and a fifth sub-question as well. But the thing is, if you don't know what the diagram represents, you're going to be at risk. Okay, anyway, the answer is plasmolysis is shrinkage of protoplasm from the cell wall when a cell is placed in a hypertonic solution. So children, remember this is a plant cell. There is the cell wall, right? You are placing it in a hypertonic solution. This is the solution in which it's placed, right? And these are, let me use a different color for the solutes to show that it's hypertonic, probably green color. Okay, right. So hypertonic, so many solutes in it as compared to inside the cell, right? Solute concentration outside is really, really high. Now what happens? According to the principle of plasmolysis, what happens is when a plant cell is placed in a hypertonic solution, the contents inside the cell wall just shrink away from the cell wall, which means they are losing water. Okay, they are losing water, and as a result, they end up looking like this. The cell wall would stay intact, but cell membrane and everything that comes within it, it just loses its shape and it just shrinks within. But cell wall is there to maintain the shape of the plant cell. All right, so this is what happens in plasmolysis. Once again, read the definition. Shrinkage of protoplasm. Protoplasm refers to the entire living contents of the cell, right? Shrinkage of protoplasm from the cell wall when a cell is placed in a hypertonic solution. Well, I would say when a plant cell or a bacterial cell is placed in a hypertonic solution, because we are not talking about animal cells in this context. Now, an example, an application of this in our everyday life, preservation of jams or even pickles, making pickles using a lot of salt. These are all methods of preservation by adding sugar, fish, meat, and all of those, like preservation of fish, meat, and all of those by adding salt to them. Pickling is what we call it, because this creates a super concentrated or a hypertonic medium, which means the microorganisms are going to be killed. Okay, bacterial growth would be prevented, even saline gargling for that matter. I would just write that also here. Saline gargling, saline water gargling, right? For a throat infection. This is where it helps. For a viral infection, it may not be that helpful, but since you may not normally know whether it's a bacterial or a viral infection, it's always advisable to do saline gargling, because it's more or less a pretty much a harmless process. Okay, right. So salt water, because you're gargling it, the bacteria, if any, which are growing in your throat, they end up losing their water in their cell contents by plasmolysis as a result of the saline environment that your salt water is creating. And as a result, the bacterial cells shrink up and they can't exist anymore. Right? All right, so let us move ahead. Here is question 9 for you. The given figure is the diagrammatic representation of an RBC, a red blood cell, kept in 3 different kinds of solutions. Name the 3 solutions in which the RBCs are kept. What changes take place to the RBCs after some time? Give reasons for the changes. Think about the answer. I'll come back to you. First one. Okay, you're given 3 RBCs in 3 different shapes, A, B, and C. I think the easiest way is to figure out which one of them is the normal shape, is the regular shape of RBCs. That will make it much easier for you to identify and work out your answer accordingly. So, children, if you actually observe it, RBC, what shape is it normally? Biconcave, right? So, a donut shape or a biconcave shape is what you see in option B or RBCB. So, this is the one which is still intact without any change in shape. So, obviously, this has to be an isotonic solution, right? Okay, first one. What do you think it is? Swollen up. When would an RBC swell up? Only when it's placed in a dilute solution, which is a hypotonic solution. And the last one, wherein it's shrunken down, that happens when the RBC is placed in a highly concentrated solution, which is hypertonic solution. Okay, now what changes would take place to the RBCs in some time? I guess the answer is there anyway in front of you, but you need to give reasons for these changes. So the answer can be organized this way. First of all, the solutions in order. Hypotonic for the first one, isotonic where there is no change, hypertonic where it's shrunken down. Now, what are the changes you would see? In a hypotonic solution, you would see that the cell enlarges, swells up, may even burst after some time. In the second one, in an isotonic solution, cell shape and size remain pretty much unchanged. Iso, equal. And in the third one, cell shrinks in size because it's in a highly concentrated solution. Then, now coming to the reasons for these. Well, the first one, the cell was kept in a hypotonic solution. So what happens is water enters into the cell and it enlarges. Right? So, like you see here, water has entered into it and it enlarged. Right? So, this is the reason for it. For the second one, the concentration inside the cell and outside were pretty much the same, which is why no change was observed. And in the third one, water was lost by exosmosis. Here you can actually write it would be endosmosis, right? Endosmosis. I think it wouldn't harm to mention that term over there. In this case, it would be exosmosis because water is moving out of the cell, right? So this is the reason for each of the changes that you would see. Remember, children, to answer this, you must know a couple of things. First of all, what is the normal shape of an RBC? If you don't know that, then you're going to mess up the entire thing. Secondly, what is the concept of osmosis? Right? These two things, if you know very clearly, and what is tonicity, the concept of tonicity, you are all well set. Okay, now coming to another question, which is write an experiment to demonstrate transpirational pull. What do you think it is? Transpirational pull, we've discussed this already, and the diagram makes it a little easier for you to understand. So you are expected to write an experiment. Children, well, I would say experiment-based questions from this are very important. From the plant physiology chapters are very important. So it can either be in this manner, but more commonly, you could expect questions being asked in the form of a diagram. A diagram being given to you and you have to answer a set of questions based on that, based on one of these experiments. So basically it means you must know all these experiments very, very well, inside out. Okay, right. So you fit a leafy shoot in a vertical tube filled with water, keep the lower end of the tube in mercury, keep the setup undisturbed for some time. It's seen that mercury will rise up in the vertical tube to a certain height due to transpirational pull. All right. So I think this is a very, very simple experiment that can be done. So understand this in detail. If there's anything else that you would like to add to this, please mention in the comment section below, because it's going to benefit many other children also out here. All right, children. So here is my homework for you for today. Give 2 examples of turgor movement in plant. Okay. Turgor movement. Put them in the comment section below. I will get back to you with the answers in the next session. And yes, children, there we go. Today we've discussed all of this. Absorption by roots, imbibition, diffusion and osmosis, plasmolysis and deplasmolysis, absorption of water and minerals, and also about active transport, passive transport, and ascent of sap. We're done with all of these major concepts. So children, yes, we come to the end of the session, but remember to note the timetable. This is our current session. We are done with this. The previous session was on the 10th of Feb at 5 p.m. It was on the chapter Structure of Chromosome. And our next session is going to be happening tomorrow, which is at 6 p.m. tomorrow. The chapter will be Transpiration, yet another plant physiology one. All right, and guys, remember the coupon code for Vedantu Pro subscription, AMBPRO. Okay, so avail the best offers that we have out there. And children, click on the like button if you have been enjoying the Sprintex series, and particularly you have found this session useful. And please, 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 children, do share it with all your friends, because as I always tell you, sharing is indeed caring, and it's a very good value. We want as many children as possible across our country and across the world to benefit from all the hard work that we do on this channel, Vedantu 9th and 10th English. All right, guys, so stay subscribed yourself and get in touch with me for anything that I can help you with. And until we meet again, stay happy, stay healthy. This is Ambika signing off. Bye-bye.